Välkommen tillbaka till Småbolagsdagarna. Let's talk about the cancer-free survival because that's the target for our next company. Please, a warm welcome to Mendus. Is that how you pronounce it?
Yes.
Yeah. And the CEO, please introduce yourself.
Thank you.
Thank you.
Hi everybody, thank you for coming out on a very sunny day to listen to small cap companies, including us, talk about their ambitions. My name is Erik Manting, I'm CEO at Mendus. Mendus is based here in Stockholm with our headquarters. We're also having offices in Gothenburg and our lab is based in Leiden in the Netherlands. We have the ambition to change the course of cancer treatment. And the reason we say that is today the biggest cause of cancer-related deaths is actually recurrence of the tumor. So it's not the first treatment, its first treatments are generally successful, but it is when a tumor comes back and it gets very hard to treat when cancer patients die, basically. And what we have developed is an approach which is called cancer maintenance therapy, whereby we try to keep patients longer in a disease-free state. We are a listed company, our disclaimer is also on our website. So we are pioneering cancer maintenance therapy. What does that mean? It means that very often after first treatment, from a clinical perspective, patients have successfully achieved a complete remission or a clinical remission, but there are still residual cancer cells. And in some tumors, that is actually causing a very fast recurrence. And one of the tumor types that we treat, acute myeloid leukemia, is so aggressive that generally speaking, the tumor comes back within a few months. And the majority of patients, if untreated, have the tumor back within the first year after the chemotherapy. So very high unmet medical need. Same thing for ovarian cancer, where we now have a phase 1 trial ongoing. And next to that, we have two other programs. One is an intratumoral primer, which is a compound we inject into solid tumors to make that tumor more susceptible to the immune system. And also we are developing as a preclinical program, a program around so-called natural killer cells or NK cells, which also have a lot of promise in the treatment of cancer. This is how our pipeline looks. The Advanced 2 trial is a monotherapy trial, so it means that we only test our compound. And this is a trial, a phase 2 trial in acute myeloid leukemia, whereby we try to keep patients longer in a disease-free state after they have received their first chemotherapy. And we have in a similar setting the Allison trial, which is in ovarian cancer. I will show you the data later, but the readout of the Advanced 2 trial last December was successful. So we are now also preparing for a follow-up trial in that indication in combination with standard of care. Then we have the intratumoral primer called Elixadencel. It has been tested in a wide range of tumors, which were very difficult to treat with other methods. And it showed clinical effects in a lot of different indications. So there the challenge was to find an indication where we think it will really be a clear-cut signal. And for that we have chosen soft tissue sarcomas and more particularly also gastrointestinal stromal tumors, which is a subgroup of soft tissue sarcomas. So with that product, we will do a decisive trial to show whether it holds up in such a tough to treat tumor. And then we have the preclinical NK cell based program, which is also based on our own technology platform around dendritic cell biology. So the indications we treat are all high unmet medical needs. In acute myeloid leukemia, the new frontier is really maintenance therapy. So again, initial therapy is very often successful, but if a tumor comes back within a few months' time, you can't really speak about the clinical success, right? So to keep patients longer in a disease-free state is crucial in this disease. Same thing for ovarian cancer. So women are treated with surgery and chemotherapy. The majority of women have the tumor back within the first year, and then it gets very difficult to treat. Finally, in GIST soft tissue sarcomas, after first-line treatment failure, basically these tumors don't respond to any other treatments currently available. So that's also one of the reasons we try to make a difference there. Our products are based on dendritic cell biology. Dendritic cells play a crucial role in the immune system, but to understand the biology and to translate it into products that are not only having a therapeutic effect, but also from a company perspective that can be manufactured, That is what we specialized in. And both of our products are so-called allogeneic products, which means that we are not dependent on patient material. The first product, Vididencel, is made from a cell line. The cell line is ours. It's a proprietary cell line. That's the start of a manufacturing process. It's actually a cancer cell line, and that sounds a bit strange, but of course the final product is safe. And what we do is we grow these cells in large quantities, and then we give them certain compounds called cytokines, and then they start to express dendritic cell biology. And that makes them, from cells that are very poorly recognized by the immune system, very immunogenic cells. And then they start triggering all kinds of immune responses, and this makes the basis for a very potent vaccine, which we are now testing as a cancer maintenance therapy. The other product, Elixadencel, is from healthy donor material. So we get leukocytes from healthy donors, we isolate a certain cell type called monocytes, and we train them to look like inflammatory dendritic cells, which we inject into tumors. That biology is something that we specialized in, and we believe that on the combined platforms, we are one of the world leading parties in understanding that type of biology. Now, what's the medical need? This is a graphic representation of what maintenance therapy is about. So an initial treatment will push the disease down, but if there's any residual disease, it will come back very quickly. And what we try to do is to, with an immunotherapy approach, basically improve immune control over the residual disease. This is just an overview of the AML treatment landscape. What it roughly boils down to is that 50% of patients undergo high-dose chemotherapy. The other half of patients are usually too fragile to do that, and they get what's called a low-dose chemotherapy. They are the chemo-unfit patients. And what is new is maintenance therapy. And there's now only one drug approved recently, which is an oral formulation of a chemical drug called azacitidine. And I will show you the data of that drug. That gives a little bit of relief for patients, but it also still leaves a lot of room for improvement. And particularly immunotherapies, which have the potential to result in very durable responses, is a big promise if, of course, successfully developed. And that's what we are aiming for. There's one additional element I want to make you aware of, which is called MRD, or measurable residual disease. So nowadays, with sensitive molecular techniques, you can pick up cancer cells in the blood of patients. In AML, it's really bad if you pick up MRD. Yeah, so this is, for example, a very big historic study showing that MRD positive patients have a really poor prognosis. The other part, which is element which is important in this slide, is the statement by Dr. Platzbecker, who is one of the key opinion leaders we work with. Maintenance therapies have to be safe. If the initial treatment is already very harsh, you can't keep patients healthy by throwing more chemotherapy after them. You have to have something else, and which is why we believe immunotherapy holds big promise in this maintenance setting. This is the drug I mentioned earlier, oral azacitidine, and the phase 3 data that led to the registration of this trial. The blue lines are placebo, the red lines is the azacitidine. And what you can see immediately is that there's only very limited and temporary relief in overall and disease-free survival. And if you zoom in on the bottom right panel, this is disease-free survival. What you see is that after the initial high-dose chemotherapy, if patients are still MRD positive, the disease comes back for the majority of patients within 3 months. So the median overall survival is 2.7 months. With azacitidine, this shifts to 7 months, but still you see that all patients in the end get sick relatively quickly. Now what we do is we give vaccinations. So we make the product Vididencel that I just showed. It's cancer cells that we reprogram to look like dendritic cells, which makes them highly immunogenic. We irradiate these cells for safety purposes, and they're being administered as an intradermal injection. So they're being injected in the skin. These were initial data we presented in May of last year, and this shows the effect on MRD. And what we saw in the initial phase of the trial is that 6 patients got sick very quickly. So we were basically not in time, or they responded poorly to the therapy. But 14 patients actually showed either a stable MRD, which is unusual because usually MRD leads to a very quick relapse of the disease. But also 7 patients showed a complete disappearance of the MRD, or an at least 10-fold reduction of the MRD. So this was an encouraging first readout. But more importantly, we could show in December of last year that it actually resulted in survival benefit. And what you see here is disease-free survival, and what you can see immediately is that this is a completely different picture from what I just showed for the oral azacitidine. Here you see that after the active study period of 70 weeks, the majority of patients was still alive. And more importantly, when we did the long-term follow-up of these patients, which is a range from 16 to 48 months, all patients remained disease-free. So this was a very good readout. Again, monotherapy. So for us, this is a very strong signal that we now want to pursue in the continued development of this compound. Also very importantly, it's very safe. The only side effect we have seen with this product is where we inject it in the skin. There's an immediate inflammation, which means that there's a big red spot. If we take biopsies as part of the study, you see that there's a lot of immune infiltration, so immune cells basically recognizing the vaccine and digesting it and training the immune system. But that redness disappears after a few days, and it's the only side effect that has been associated with the product. So very safe product. What do we want to do next? This is basically a graphic overview of the AML treatment landscape, where you see that half of the patients are so-called chemo fit. They get high-dose chemotherapy, and afterwards they either get a transplant, so they get the blood system basically from a donor, or, and that's for the major part of patients actually, they are not eligible for a transplant, or they can't find a transplant quick enough. And then there is nothing except for the oral azacitidine, which has recently been approved. So that is the main focus for us. That was the current patient population we treated. So our next trial will be to combine with the oral azacitidine in this specific setting. But there are ways to broaden the positioning of this drug. Also, after transplant, very often MRD leads to relapse. Actually, relapse is the most important reason for transplant failure. So that could also be a setting to position our vaccine. And the same thing for the chemo-unfit patients, which until recently had very poor initial response, but now with a new drug called venetoclax, do show more complete remission rates. So also there we believe there will be a maintenance population that we can treat with our vaccine. But we start with the most imminent patient population, which is the combination with oral azacitidine as a next trial. What we do as part of our research is, for example, test in animal models if our drug can be combined with other currently available drugs. And in this case, we did a combination with azacitidine and venetoclax. And what you see here, and particularly the red line is the most important one, that's the combination of our vaccine with the azacitidine venetoclax combination. You see that it's actually synergistic, so it has an extra effect if you combine these drugs. So this gives us the confidence that we can develop our product more broadly in AML maintenance. So what's the plan? The next study will be a combination with oral azacitidine. That will be a control arm trial. And dependent on the results, we can either go into a full-blown phase 3 registration trial, or if the effect is big enough, let's say similar to what we've seen in the phase 2 monotherapy study, we can translate those data into a pivotal stage trial design and potentially an accelerated route to approval. How exactly that will play out is at this moment not clear yet. We need to have the data from the first part of the trial. We need to talk to regulatory agencies. But the first part of the trial will be a combination trial with azacitidine as a step up to pivotal stage development and in the end market registration. Then wrapping up, this is the setting in ovarian cancer. Again, a tumor that is successfully treated initially but comes back very quickly and then becomes harder and harder to treat because it becomes insensitive to the initial line of treatment. So there also we try to prolong the disease-free survival. That trial is ongoing. So far the data were positive. So safety data are good, feasibility is good. We see immune responses triggered by the vaccine. And the trial design is roughly the same, so 6 vaccinations and then followed up by the monitoring of patients. This is about the intratumoral primer. It has been tested in a lot of different diseases. So for us, the main challenge here was to find a disease area where we think we can make a clear-cut difference. And we've decided to go after soft tissue sarcomas, which are very poorly responding to other currently available cancer therapies, including other immunotherapies. And to a certain extent, that is based on the initial signs of efficacy we saw in this group of tumors, which is gastrointestinal stromal tumors. And what you see here is patients that have undergone multiple lines of previous treatment, had become resistant against those treatments, and then 2 out of 6 patients started responding again after we had introduced our product. So this is a starting point for a trial that will have to deliver so-called proof of concept. And then as a last part of our pipeline, we found out that the cells we make out of our platform can not only be used for vaccination, but actually also to train immune cells outside the body. And what you see here, the middle panel, so the panel on the right of those two graphs, is that we actually found out that so-called memory NK cells, which have a high correlation with survival benefit in cancer, respond very strongly to being exposed to our cells. And this means we can make therapeutic quantities of these specific memory NK cells, and that's a very interesting starting point for NK cell-based therapies. And that can be either the cells alone, or it can be the cells in combination with antibodies. And there's now a lot of antibodies being tested in combination with NK cells. So we think this is a very promising first step for us to also enter into this NK cell field, either by ourselves or in partnership with others. So to sum it up, most importantly, we have positive data from our AML phase 2 monotherapy trial, with additional data expected, survival data expected in the fourth quarter of this year. We will continue to develop that program in a new trial in combination with oral azacitidine. And we have the possibility to expand more broadly into the AML maintenance setting. We, of course, have the phase 1 study ongoing with ovarian cancer that will also deliver readouts in the second half of this year and early 2024. And we are about to enter into a proof of concept trial with ilixadencel. And again, the memory NK cell is something that we continue to develop in-house, but which we will also look out to partner with other companies. With that, I'd like to hand it over for the Q&A session.
Thank you so much, Erik. You are based in the Netherlands, but how come you're listed in Sweden?
Well, it had a scientific basis. So we specialized, when I started in the lab in the Netherlands, in so-called allogeneic dendritic cell biology. So we know how to work with dendritic cells and how to work with, let's say, off-the-shelf platforms, which are important from a manufacturing and from also a therapy perspective, so that we have the product on the shelf ready to be delivered to patients, instead of being dependent on patient material. And then we found out this company in Sweden, Immunicum, was working on the same basis, with another source of material, with healthy donor material instead of a cell line, but with very similar concepts. So I got together with the CEO at the time, now our CSO, Alex Kalsampara, the founder of Immunicum, and we connected very well, and we immediately understood we had very similar ideas about how this kind of products work. And that was the basis, actually. So a few months later, actually end of 2020, we combined both companies and we renamed the company Mendus in 21.
And you were earlier the CEO of DC Prime, and then you took over the new company, so to speak. How come that you were the one that was appointed CEO and not the former CEO of Immunolog?
Well, that you have to ask our board. That's not up to me to decide.
Yeah, what is your mission?
My mission is to realize our slogan, which is to change the course of cancer treatment, and that's what we go for, and everything else is subject to that goal. In that context, we announced last week a financing transaction, which is a combination of a rights issue and a directed placement, but also an alliance with a Swedish company called NorthX. NorthX is a very ambitious biologics manufacturer. And we found out that the facilities they currently have and also their expansion plans very well fit with our ambition level. So we now put together this as an alliance to set up a manufacturing facility that will also allow for large-scale manufacturing of our product, which is of course extremely important for the late-stage development. So to cut a long story short, now that's where my focus is in day-to-day life. And
NorthX, you announced, as you said, a few days ago, the collaboration between Mendus and NorthX, and they are based not in the Netherlands, but they are based in Matfors, right?
Yes.
Yeah. Is that where the production will take place as well, or?
Well, we've decided not to say too much about it. I think NorthX has a high ambition level, but that is, put it simple, the Matfors facility is a very good facility, and it will serve as a basis to start the relationship.
So when do you think the production can start?
Immediately after the summer. Of course, we hope to have the money on the bank in August, and then in September we start. The facility is nearly perfect. We just need to make a few adjustments to make it suitable for our product, but the most important part is the teamwork. So what this is all about, any manufacturing process, and we got some questions about this too, like what are you going to do with the investment? Are you going to build a new factory or whatever? No.
Yeah.
Actually, the factory is only
It's
a very small part of the total investment because it was already nearly perfect for at least the start of our manufacturing process. It's really about teamwork. You need to train a team in a GMP environment to work with your cells, to work with your process, to make sure that you go through all the different training and qualification runs until you can make the product on a GMP level for pivotal stage development and in the end commercialization. So the investment we have now agreed to with Fleury, who's also a shareholder in Northex, will cover that whole trajectory up to the point where we make the first batches for pivotal stage trial manufacturing. So we're very happy with that alliance. And again, it's teamwork. It's really the conviction that we can here in Sweden establish a manufacturing infrastructure that is not only high quality, but also suitable for our late stage and in the end commercialization.
So Fleury, as you mentioned, they are also shareholders in Northex. But what will their contribution to your company be aside from providing money?
Well, they're a very renowned investor. You know, they don't take easy investment decisions. Also, of course, the founder of Fleury, Thomas Eldered, he has a lot of experience in manufacturing out of his Recipharm time. They have a very high ambition level to also become a large and growing player in cell and gene therapy. So it's a perfect match, I would say.
Okay. And this investment is expected to cover all the activities until Q2 2025. Can you tell us a little bit about your long-term financing plan? How will you finance the operations after that?
Well, any biotech development goes from value inflection to value inflection, right? And you have to make sure that you stay financed, but at the same time also that you're financed to a point where you can reach the next value inflection. So for us, the most important part now is to be ready for pivotal stage development. And that means we have to do this combination trial with azacitidine. It means we have to have the manufacturing ready for late stage development and commercialization in the end, because it's the same process. So that is really the first big hurdle we are now able to take. And then, of course, everything else in the meantime can happen. And that can be a next round of financing. It can be a partnership. It can be anything that helps us accelerate and broaden the development of the product. But this is a very important first hurdle that we are now able to take.
I understand. And you mentioned, of course, the results for the positive results for vididencel. How significant are those results that you announced yesterday?
Well, the results that they were announced in December, of 2022, last December at ASH. But the relevance, I think we should always be, let's say, conservative in the sense that your next trial has to show more. But this is the second time we show it. So also in the phase 1, which was just a safety trial, we saw that the patients that responded to the vaccination had a very extended survival curve, and in the end, 36 months overall survival, which was far beyond initial expectation. And we did a very deep analysis also in the phase 1 on the profile of those patients to make sure that they were not incidental outliers or that it was not a coincidence. Now we've shown for a second time that survival benefit can be very extended if you have a successful immunotherapy. And that, I think, is a real breakthrough. When we started, there was a lot of disbelief because AML is blood-borne disease. Patients undergo high-dose chemo. So it doesn't make a lot of sense that immunotherapy would work. And actually, almost all immunotherapy attempts so far have completely failed. So we went against all odds. And now we see again in the phase 2, what we also saw in the phase 1, that if patients respond, the immune system is quite robust. So even after this first round of high-dose chemo, and we also do a lot of so-called immunomonitoring work, so we do science in patients to show what happens with the immune system, we consistently see that the immune system, even after such high-dose chemo, is still able to mount an immune response against the vaccine and basically getting improved control over the residual disease with very long survival benefit as an outcome. So yeah, we couldn't have been more wishing for more, basically, as of December last year. So then, of course, the question is, what's next?
Exactly. Thank
you.
So
the next trial is again the next trial, right? So it's all in the game. But we feel very confident.
Thank you. We'll take the last question, and that is a question from the audience. How is the Advanced 2 study proceeding, and how is it proceeding?
Yeah, so the Advanced 2 trial was first about, of course, recruiting the patients, which in a monotherapy setting is not easy, because in a disease like this, you have to basically convince doctors and patients to use your experimental drug. So it took us quite some time and 10 centers throughout Europe, including Sweden, to recruit these patients. And then we had to collect the data, and the data are, first of all, the survival data we presented last December, but also we did a lot of immunomonitoring data collection, and you do that afterwards. You collect first the samples, and then you can open up all the vials and do all the immunomonitoring. So that's the work we are publishing currently at different conferences. The next survival update will be in Q4 of this year. But of course, yeah, we feel already encouraged by the results, including the long-term follow-up of last December. So we will not wait for those data to continue to move forward.
Okay, that was your last word here today. Thank you so much, Erik.
Thanks so much.
Thanks for coming.
Thank you, everybody.
Big applause.